Well, it's noon. Shall we, or 1 rather? Shall
It
we
is.
kick it
Jeff,
off?
are you going to present Rachel for us?
Yeah. Is that okay, Rachel? Make sure she's here.
Yeah, well, she's presenting her screen. So welcome everyone to Darwin Day. I'm going to let Jeff take over, but if anyone wants birthday cake, I've got some cake in my office. So come grab a piece of cake if you're here on campus. Jeff, go ahead. You take over.
Hey, I'm Jeff Zahn, biology teacher here at UVU, and I'm really excited to present Rachel Keuler, who I met over at BYU. Last year I went to give a seminar and had a chance to meet with some of the grad students, and I was super impressed by her passion for research in lichens, which are funky critters, and only the nerdiest of people really understand how awesome they are. So Rachel, you got your undergraduate, your bachelor's degree at UW-Madison, right?
Yep.
In conservation biology, and you're now at BYU, PhD candidate studying lichens. I should also point out that Rachel's Instagram page is full of awesome watercolor artwork, and she does a lot of science communication as well. So check out her website. And with that, I think you're free to get going. I'll mute myself here.
All right, awesome. Thank you for the introduction, and thanks for having me today. I'm super excited. I love talking about lichens, and I love getting people hooked on lichens because I think they're a really special kind of organism. And so I'm excited to share some of that with you today. So I'm a third year PhD student. I'm not a PhD candidate yet, but hopefully I will be this year at some point. But yeah, I'm studying lichen phylogenetics in the Levitt lab at BYU. I guess with that, I will get started. Just let me. Okay, so people often ask me how I got into lichens. So to kind of give you a good idea of like where I came from and how I got into something that not that many people are into. So it all started during my undergraduate when I went on a class trip to Chile. We only had 10 days for the entire trip, so we had to make every minute count. We spent a few days in Puerto Williams, which is right at the bottom tip of Chile. And we were on the Navarino Island, right on the Beagle Channel, which is named after the ship that carried Darwin's journey around South America. But one major difference between my journey to South America and Darwin's is that mine was far less grueling, though I did have to spend a night sleeping on the floor of the Toronto International Airport because of flight problems, because it was in the wintertime in the Northern Hemisphere. And down at the bottom left picture is actually the Beagle Channel. We went to Omora Ethno-Botanical Park, which is part of the southernmost forest of the world. And in the few days that we had there, we kicked off a pilot study to look at how the UV exposure, because it's right under the ozone hole, we wanted to look at how UV exposure impacts lichen communities that are growing on the beech trees there. And this was my first time really seeing and learning about lichens. I had just traveled across the world to a place that was completely new and exotic to me, and I was introduced to this really funky organism. And then I got on the plane, flew 12 hours back to Wisconsin, only to realize that lichens weren't just in this exotic place across the world, but they were everywhere. They were growing on all different kinds of rocks of various types. They were growing on every species of tree that I could see. They were even growing on soil, among moss, growing on sand, growing in bright areas, but also like dark, moist areas. They were even growing on man-made objects like metal signs, buildings. The right picture is a close-up of a gravestone, and you can see that the lichens are just growing in the etched-in letters. And once it became apparent to me how widespread lichens are, I I got hooked. I had to learn more about them, and so that's kind of what kicked off my journey, I guess you'd call it, into studying lichens. First, I want to give you some of the context behind the study of lichens, and they're not as straightforward as other things that you see in nature, and so I wanted to kind of give you some context behind how we got to where we are today. So throughout most of human history, scientists have had a hard time understanding what lichens are. For hundreds of years, they were classified as some kind of plant, even though taxonomists really couldn't find any parts that resembled a plant, but they didn't know what else it could be, and they didn't really care because they don't really have a lot of economic value. So for many years, they're just kind of thrown into the plant category. And so scientists tried to find a way to categorize them. Italian botanist Pietro Antonio Micheli wrongly assumed that the asexually reproductive part of the lichen was actually a seed. And you can see a picture of that on the left. And Linnaeus, who is the father of taxonomy, agreed with Micheli, but he had some hot takes on lichens, calling them the poor trash or paupers of vegetation. During his time, Linnaeus named over 12,000 species, but just 100 of them were lichens. So that kind of indicates that he was not a fan of them for some reason. And it wasn't until 1869, just 150 years ago, that the Swiss scientist Simon Schwendener proposed the dual theory, suggesting that lichens were made up of a fungus that was parasitizing or enslaving an alga. This idea was pretty controversial among the leading lichenologists of the time, who still firmly believed that it was some kind of plant. And then 10 years later, in 1879, German scientist Anton de Bary coined the term symbiosis as the living together of unlike organisms to describe lichens. And then after that, scientists slowly, over time, started to accept that this was some kind of, it wasn't one organism, it was some kind of interaction between different species. So now, today, it's accepted that lichens are the result of a stable symbiosis between a fungus and an alga or cyanobacteria. Today, I'm going to refer to the alga cyanobacteria as the Photobiont, because it's, and that's named so because it's photosynthesizing, it's taking in the sunlight and turning it into carbohydrates or food that the fungus utilizes as well. The photobiont, as you can see in this picture, is nested among the fungal hyphae, which are the filamental threads of the fungus. And the photobiont is able to live and grow in more places because of this shelter that the fungus is providing. And you can see on the left that it's kind of growing in layers. The top layer is like this mat of fungal hyphae, and then below that, but not too far below that, the sunlight won't reach it, is that algal layer. And I'll use, in this talk today, I will use the word species to refer to lichens, but in reality, they're more of a composite species or even an ecosystem. And when you isolate one from another, the fungus doesn't form the same kind of specialized structures. Instead, it grows in these like mold-like colonies. And generally, the fungal symbionts can't live on their own without this symbiotic interaction, but many of the algal species do and are found living solo in nature. So there's over 20,000 species of lichens around the world, and 20% of fungi can be lichenized, and that means that they're able to form this symbiosis with the photobiont. And they are covering around 7% of the Earth's surface in a variety of extreme ecosystems, from the tundra to the rainforest. You'll even find them out in the desert in Utah. And There's even been studies where they sent them off to space and they survived. They were able to survive a trip to space and back. So they're really this hardy species, or like composite species, that's able to survive in a lot of different situations. And so really often I get asked, what is the point of studying lichens? And people want to know, what do they do for me? You know, I've never noticed this thing out there. I don't understand what the value of it is. And while I believe that lichens have an inherent value and they are contributing to the ecosystem and really furthering our understanding of symbiosis, the cyanobacteria in lichens can fix atmospheric nitrogen, helping it, making it usable so that plants are able to utilize it. They're also a pioneer species that can be the first to colonize a barren landscape or a disrupted ecosystem, like after a wildfire. And they can break down rocks through physical and chemical weathering, helping it make the ecosystem more habitable for future organisms. And here you can see it can be of value to many different animals as well. On the left is a green lacewing larvae carrying lichen for camouflage, which is pretty adorable, I think, and I love it. In the middle, you can see it can be an important food source for reindeer during the winter. And then on the right is a cute little nest, bird nest, and there's also a lot of pictures online that you can see where hummingbirds have made these really cool little nests out of lichens. We're also starting to really utilize lichens as an affordable way to assess air quality. Lichens can be a useful air quality biomonitor because they're sensitive to nitrogen and sulfur dioxide. So now that you kind of understand what lichens are and how they're tied to the ecosystem around them, I want to talk a little bit more about their evolution. For many organisms, the fossil record can be invaluable to help date their evolution. But lichens have a pretty abysmal fossil record because they lack the biodegradation resistant compounds, and they're often growing in these extreme habitats, these extreme ecosystems that aren't conducive to making fossils. And in this picture, you can see a charcoalified fragment of lichen from the Devonian. And I think this is super awesome because you can see the cyanobacterial cells that are nested among the fungus. And you can even see that in this really old fossil. And the oldest known lichen fossils date to the early Devonian, which is over 400 million years ago. And additionally, there's a lot more in comparison to the fossils. There's a lot more lichens that have been found in amber, in the Baltic and Bitterfield amber in Europe, which is really cool. And you can see that on the left here. And the origins of likanization, which again is that the symbiosis, the ability of the fungus and the photobiont to come together to form this structure, the origins of that are still unsettled. Some scientists believe that there were multiple independent origins of likanization, that it had evolved independently multiple times, but others suggest that it evolved once and that trait was lost multiple times. And the argument for that is that this is a really complex, kind of thing, you know, to be able to evolve this symbiosis between two organisms is it seems like a really complex kind of evolution. So it's really difficult to understand how and when that symbiosis evolved. And here is the geologic timescale with more recent towards the top and further back towards the bottom. And you can see that the lichens, as I mentioned, according to the fossil records, the lichen symbiosis emerged in the early Devonian, and lichens were, you know, hundreds of millions of years after that. And then humans are just like we're so very young in the geologic timescale. So lichens evolved much before we have, and so they've been around. They've had a lot of time to evolve and become what we see today. And so here is the tree of life showing the 6 kingdoms that we have grouped every organism into. And so on the left in the red circle is where the cyanobacteria lies on the tree. And then we have the green algae circled in green, and then the fungus in blue. And so you can see that the lichens require a symbiosis across many kingdoms of the tree of life, which I think is super interesting. And then here is the tree of kingdom fungi, and 98% of lichenized fungi come out in Ascomycota. There's a few in Basidiomycota, but the vast majority of lichenized fungi come out in Ascomycota, in just this one group in the evolutionary tree. And so while we've come a long way in our understanding of lichens since Schwendener first proposed the dual theory, there's still a lot to learn about lichen symbiosis and its evolutionary history. Lichens themselves are named after the fungal symbiont, and so much of the study of lichen evolutionary history focuses on the evolution of the fungal symbiont alone. And I would argue that this is due to 2 things. First, we're still trying to shake the dominant scientific thought that the fungal symbiont is what runs the show. And for the longest time, we believed that the fungal symbiont is really the most important symbiont in this interaction. And so their evolutionary history is what matters the most. And so, and second, the second reason why I personally believe that we just focus on the fungal symbiont is that we don't really have the technology yet to study how a symbiotic composite organism evolves over time. There's some studies that have looked at how the photobiont has evolved, but there are not very many studies looking at how a specific lichen, this like specific symbiosis of one lichen composite organism, has evolved throughout time. And today I'm going to share some of my research, which is focusing on the standpoint on the evolutionary history of the fungal symbiont. But I want you to be aware that this is only one facet of a complex symbiosis, a complex symbiotic evolution. And so I'm really excited to talk to you today about my research on the evolutionary history of lichens. So now that I've given you the background on lichens, I want to take a moment to step back and talk about evolution more broadly, even beyond lichens. So just as the study of lichens has transformed over time, so has the study of evolution. So you might remember learning in introductory biology classes that hybridization is an evolutionary dead end. Maybe you've learned that even if two different species can physically reproduce, the resulting offspring may be infertile, like the example of mules, which are always sterile, or they have such a reduced fitness in comparison to the parent species that they would never stand a chance in competing with them, and that lineage would just die off right away. And another thing that we learn in introductory biology classes is about the biological species concept. That in order for an organism to be characterized as its own species, it must be reproductively isolated. And this has been the dominant way of thinking for the past several decades. But with advances in gene sequencing and new methods of analyzing these genomes, we're discovering that things aren't that simple. In fact, gene flow between species may not be that uncommon after all. And so I want to point out, so the things that we're learning, there's a lot of research that's been going on about the hybridization between humans and Neanderthals. And maybe you didn't know that hybridization may have even affected your genome. There's studies that have found that all non-African humans carry traces of Neanderthal DNA due to early hybridization events. And you can see this in this figure on the bottom left, where long ago Neanderthals and humans have hybridized with each other, and we're finding so much evidence of that in the human genomes, which is really fascinating. One group of researchers in 2011 studied the human Neanderthal hybridization and developed an analysis that allows researchers to detect these ancient hybridization events without having to have ancient samples. So you don't need a fossil or some sample that had been preserved in the glaciers or whatever. You're able to use the genome from something that is alive today, and with this analysis, you can kind of see back in time. And so this analysis is called D-statistics, or ABABA. And I will bring this analysis up again later because I was able to use this also in my own research, which is really cool that you can use something that was developed for this interaction and apply it to something to do with lichens. And so now I want to introduce you the Taco Bell analogy. And this is something that my advisor, Dr. Levitt, mentioned to me, and I absolutely love it. This is not sponsored by Taco Bell, but here we have a picture of two Taco Bell menu items. One is a taco with repurposed Doritos as the shell, and the other is like this combination of, I would say, a quesadilla and a hard shell taco. So rather than putting effort into bringing in entirely new ingredients to create entirely new menu items, Taco Bell repurposed these ingredients that already exist across the menu, putting them into new forms that they can say, hey, look, we have a new menu item, let's try it. And evolution, believe it or not, can operate on similar mechanics. So while evolution is often driven by new mutations, this can be a really slow, ungainly process. And on the other hand, speciation through the reassembly of old genes, so using these genes that are already in the genome, they're already across the population, they've already been tested by selection. So the reassembly of these genes that have been introduced by hybridization can really help populations evolve a lot quicker than having to develop than like randomly developing a mutation and passing that mutation around the entire population. So an example of this Taco Bell evolution is in action with the cichlid fishes in Lake Victoria in Africa, and also Darwin's finches in the Galapagos. So first, I'll go into some of the research with the cichlids. So there has been a ton of research, like this is a really well-studied organism, looking into how cichlids were able to evolve so much variation in such a short evolutionary timescale. And they were able to evolve so much variation over just 150,000 years. And so some research suggests that the genetic material exchanged between two divergent lineages, so the Upper Nile cichlids and the Congo cichlids, is what fueled this rapid radiation that was able to bring in so much variation in such a short amount of time. Another well-studied area of adaptive radiation is with Darwin's finches in the Galapagos Islands. Charles Darwin first noted the wide diversity of beak shapes on the Galapagos Islands, and now research using their genomes has revealed that, like with the African cichlids, that an early hybridization is what was bringing in the genetic diversity that was able to fuel such a wide diversity in beak shapes. So it's really cool that back in time, Darwin noticed this like really special, novel thing that was happening with the finches on the Galapagos Islands. And now with our current technologies, we're able to say, yeah, this has like a genetic basis, and there's a reason why this is happening. And it has to do with this like genomic material that has been introduced by hybridization. So now I want to shift a little bit into my research. Hybridization among plant pathogenic fungi has been well documented. They're a lot easier to test in the lab compared to lichens. And there's a lot more funding going into this because, as you might imagine, we want to be able to protect our crops. There's an economic value for it. So a lot of research has gone into looking at how these pathogenic fungi have hybridized and almost like created new strains that are able to be more aggressive on crops. And so it's important for us to understand how this process works in fungi. And hybridization with other fungi is just absolutely lacking. There's not really been a lot of research looking into this. And with lichen-forming fungi, only a handful of studies have detected it or even considered looking at it. And it makes sense because lichens are a tricky organism to work with in the lab. It's not really as easy to experimentally test their reproduction in the lab. And so we're really limited by how we can look into evolution or look into this process of hybridization between lichen-forming fungi. And so in my research, I've started applying some of the same analyses that have been used in the Neanderthal and cichlid studies, applying them to lichen-forming fungi to try to get a better understanding of their evolutionary history. This project started because of a speciation and phylogeography class that I took my first semester in grad school. And since my advisor taught it, we looked at lichens for the class project. And after the class finished, my classmates, which were a bug guy, a fish guy, and a plant lady, were just pretty much over working with lichens. They didn't want to deal with an organism that is just this intricate mess of like relationships between different organisms, and it's just too complicated and too messy. So they didn't want anything to do with it. And I took over in wrapping it up. And at the time, I had no idea what I was getting myself into. At the time, I didn't think that this is what I was going to devote my PhD research to, but now I am, and I absolutely love it. And I think it's a great thing to look into with lichens. And so I'm super excited to share it with you today. So my research looks at vagrant lichens, which are lichens that grow unattached to any substrate. So usually lichens are anchored onto something like a rock or a tree, but these lichens are unattached. They can move around with wind or water, though they don't really move around that much because they're kind of just like tucked away between these shrubs. And but they're often found in open, windswept habitats around the world. In the western United States, they're often found in the cold steppes. And these vagrant lichens are rarely sexually reproductive, so they're often vegetatively reproductive, which means that a piece can break off and then that's a new lichen. I know that seems like that's not a form of reproduction, but that really is how these asexual vagrant lichens asexually reproduce, is just breaking off a piece and then establishing elsewhere. So my lichen, for my study, I wanted to look into the potential role of hybridization among species in the rock-posy species complex. Species in this group are found worldwide in mountains, deserts, even Antarctica. And so on the left, you can see a picture of what the group is made up of. So the rock-posy group is made up of rock-dwelling sexual species, which I have pictured on the left, and is also made up of vagrant asexual species, which is the picture on the right. And these species often co-occur in similar ecosystems. So since hybridization can generate novel traits and reproductive barriers in fungi, I wanted to look and see if I could detect hybridization in these vagrant asexual lineages. So again, to reiterate, I wanted to investigate the potential role of hybridization in the rock-posy species complex, looking at how it could have potentially affected the evolution of these vagrant forms and also influencing the differences in reproductive strategies, the asexual versus sexual. For this study, I sequenced 55 specimens within the complex. I sequenced both the nuclear and mitochondrial DNA. In the end, we sequenced 20 million base pairs and generated 407 genes. I used 3 different methods of tree reconstruction. I won't go into detail about these analyses or why I used them, but the overarching reason why I used 3 different methods is because each method operates on its own assumptions and has its own strengths and weaknesses. So when you use 3, you can compare and contrast the results to get a better understanding of the evolutionary history. Every analysis that I did for this project had to be done on BYU's supercomputer because regular computers just cannot handle that much data. They don't have the processing power that is necessary for these types of analyses. So I learned how to use Linux and the supercomputer, which is, for me, a pretty steep learning curve. And I had a lot of help from BYU bioinformatics undergrad Hayden Smith, who is a genius. I also used 2 different tests for hybridization. I used PhyloNet, which tests and visualizes where potential hybridization could have happened. So instead of giving you a bifurcating tree, which is a tree that is just branching off into 2 species with each branch, it returns a network, like you can see at the bottom with that phylogenetic network. And which is great because I think this is one of the first programs that really has given us the ability to generate a phylogenetic network rather than just a simple evolutionary tree. And then second, I used D-statistics, which, as I mentioned, oops, I don't know why that slide just went forward, but we're going back. And as I mentioned, D-statistics is an analysis that was developed to assess the relatedness of the Neanderthal genome to present-day humans from different countries. And this method specifically assesses gene flow in the genome, and it helps you identify which parent species may have contributed genetic material to produce which hybrids. So here is the resulting phylogenetic tree that shows the evolutionary history of our species based on the DNA that we sequenced. Highlighted in red is the vagrant asexual group. If you've never seen a phylogenetic tree before, don't worry. All you need to know is that all 3 of our tree reconstruction methods generated identical branching patterns with really good statistical support. This amount of similarity between these methods is a great sign because it shows that our data is robust. And most phylogenetic studies would just leave it here and call it a day, say that, you know, we have these great trees and this is the evolutionary relationship based on the DNA that we sequenced. But this figure, which I understand is also very complex, so let me walk you through it. This figure is comparing the evolutionary tree generated from the nuclear DNA, which is the tree on, maybe I can actually get my laser pointer. So this tree on the left is the tree that was generated with the nuclear DNA that we sequenced. And then here on the right is the mitochondrial DNA, the tree that was generated from the mitochondrial DNA. And then highlighted in green right here is the vagrant asexual group. In red, right here and here, are the 2 instances of mitonuclear discordance, which is where the nuclear tree and the mitochondrial tree are disagreeing with each other. And normally you would expect to see the same history between each of them. But when there is this discordance, it can be evidence for hybridization. Sometimes things get jumbled up during hybridization, which more often than not can lead to incompatibilities and reduced fitness. And so more simply stated, it's not great if it gets jumbled up like this, and it doesn't really help anybody. But sometimes, just sometimes, it can jumble things up in a way that generates new physical traits. So what this figure tells us is that it's possible that this vagrant asexual group was involved with in a hybridization event. And the D-statistic test, which isn't pictured here because there's not really a great way to visualize it, but that test also showed evidence of hybridization involved with the vagrant clade. And here is the phylonet results showing one reticulation or one hybridization event. And it's pretty straightforward to interpret, right? So the orange branches show that Rhizoplaca melanophthalma hybridized with Rhizoplaca pirilis to give rise to this new lineage. However, statistically, this model wasn't the best fit for our data. So here's the result of two hybridization events. It's a bit more complicated, showing that there is gene flow among several different species, but this model also isn't the best fit for our data. So this model, using 3 reticulations, was the one that was our best fit for our data statistically, although it's possible that more reticulations could have been an even better fit. But any attempt that I made to run an even greater number of phylonet reticulations was too much for the supercomputer to handle. It just would not, it said, nope, you are not going to do more reticulations. That is too much. So this isn't like the first model that I showed you. It's not like this one where I can explain it in one sentence. It's for example, if you look at this, we have this ghost lineage, you know, it's coming from all the way back on the backbone, something that was around before now, sometime in the evolutionary history. This ghost lineage hybridized with this other ghost lineage to give rise to Rhizoplaca melanophthalma, and like, it's just like, how do I explain this? And in any case, while I was writing up the scientific manuscript for this study, I did everything that I could not to have to put this into words, because I don't even know how you would describe this scenario. All you can really say is, yes, there was gene flow happening among the species in this group, and yeah, that's all that we can really say about it in interpreting this tree. So here you can see Darwin's famous tree on the left that was originally published in his Origin of Species. So while Darwin's conclusion was revolutionary at the time, we now have the tools available to help us visualize just how complex evolution might be. What if evolution can't accurately be depicted by a branching tree, but is something like the network on the right? Or even more complex, like a spider web, where everything is kind of interacting with each other. It's not this simple linear evolution. So for my rock posy study, we originally hypothesized that hybridization had played a role in the evolution of vagrancy within this rock posy species complex. We found evolution of this with the mitonuclear discordance, that discordance between the mitochondrial DNA and the nuclear DNA, and these different tests for hybridization. So this study highlights the value of looking into hybridization, even in cases of well-supported phylogenetic trees. Though I want to mention again that my study only looked at the evolution of the fungal symbiont, which is only one piece of this symbiotic puzzle. We missed the story of the Photobiont as well as the symbiosis as a whole. And so science is about trying to gain understanding in an infinitely complex world. We have these complex phylogenetic networks, complex symbiotic organisms, and I would be more than happy to be proven entirely wrong in 150 years if it means that we have a better understanding of how such a beautifully complex symbiosis works and evolves together. Learning more about lichens brings us even closer to understanding how the world we live in operates. One of the reasons it took scientists so long to understand that lichens were a symbiotic ecosystem is because they lacked the tools to understand that. And one of the reasons that we spent better part of a decade, or better part of the 20th century, thinking that hybridization led to a dead end, is because we also lacked the tools to detect that interaction. So with new technologies, scientists are just now starting to see how common hybridization or gene flow between species might be. Just like Schwendener was met with resistance when he proposed the dual theory about lichen symbiosis, for decades, a lot of scientists, well, some scientists have been fighting an uphill battle to get others to recognize the relevance of hybridization in evolutionary histories. So only now, with our increasing ability to sequence and analyze genomes, we're beginning to unveil the tip of the evolutionary iceberg. Hybridization has now been detected among a wide variety of plants, fungi, and animals. And here are pictures of some of the most well-studied groups, the heliconius butterflies. These are the cichlids again, sunflowers. And so we're able to really dig into these genomes to understand the complex evolutionary history. And I just want to say it's important to recognize that science itself is also constantly evolving, just as the organisms we study are. Science is largely influenced by the technologies that we have and the society that we live in. Charles Darwin came up with a theory of natural selection without even knowing what DNA is, which is pretty cool if you think about it. And who knows what ideas we have today that will be backed by the technology of tomorrow. And so this is why we shouldn't be discouraged by our increasing understanding of the complexity of life. People resisted Darwin's theory because it made the world more complicated. And people resisted Schwendener's theory because it made lichens more complicated. But in comparison to what we know now, these ideas seem simple. And lichens are a part of a new evolutionary frontier that's being paved by these new technologies like gene sequencing. And I myself am often overwhelmed by the thought that evolution may be too complex to ever really truly understand. But in our quest to understand the world we live in and what has shaped it, I've gained a much deeper appreciation for life as it is and as it once was. So I just want to say thank you for listening to my talk today. I was really excited to be given the opportunity to present, and I really hope that you came away from this with the same awe and appreciation that I have for lichens and evolution. So thank you.
Thank you.
We've got some time for questions now.
I have a question.
Sure.
Do you have a feel for, we just saw a seminar yesterday by a job candidate who was looking at the relationship between fungi and root nodule formation in plants. Is there somebody looking at the signaling, the communication between the fungus, the fungal portion of the lichen and the photosymbiont portion of the lichen? And who is?
Oh, go ahead.
I'm just wondering where the references are, where to find out what the state of that is.
That's a really good question, and I don't know if I don't think there's that many people looking at that. I think that's a really great idea because they are living so closely together, and there must be some communication between them. But I'm not sure how much research has gone into that. But that's a great question.
So one of the hardships of that type of research is if lichens grow as slowly as I think they grow, it might take a long time for evidence of communication to emerge, and the signals might be vanishingly scarce because it would be an accumulated signal over time. What are the fastest growing lichens and how fast do they grow?
I'm actually not sure. So some lichens, I think especially, so crustose lichen, well, maybe I shouldn't say things that I'm not entirely sure of, but some lichens do grow very, very, like very slowly, but there are faster growing lichens. And you can kind of figure that out based on what they're growing on. So if it's like a younger tree, you can kind of get an understanding of like, oh, okay, this established here recently. But I'm not sure off the top of my head what's the fastest growing lichen.
Okay. Well, I knew it was I didn't mean to put you on the spot, but
oh no, it's okay.
That's the thought that came out. Thank you.
Mm-hmm.
It looks like Sophia's got her hand up.
Go ahead and ask your question, Sophia.
Okay, sorry, I just don't want to interrupt if someone else had a question. I recently found out about lichens being sold commercially, but the problem with this being is because they grow so slowly that they take them out of the environment to, sell them. I was just wondering if you knew anything about like the heavy effects that it's having on the environment, since it's so necessary for, like you said, like reindeer and other organisms to use for their own survival.
Can you repeat your question again? Sorry, it was a little bit unclear.
If you in your research, if you had like researched any of the effects that commercially sold lichens had like on the environment.
Yeah, I haven't seen much research looking into that, but that is definitely something that like you're starting to see more and more where like people are going out and collecting. Like we don't really have, lichens aren't something that we can easily grow. So anytime that you see these lichens that are being, I think the first thing that comes to mind is that you can get them bought, like you can buy them in bags at craft stores, but they say they're a moss, but they're actually lichen. But like those have come from, those have not been grown sustainably somewhere. They have come from the ecosystem. And so I do wonder what kind of effects that has had on the ecosystem, but I'm not sure. I'm not sure if it's been something that is really, I'm not even really sure where they're harvesting them from, and if it is from these ecosystems where, like you said, reindeer are relying on them to get through the winter. But that is a great question.
Thank you, yeah.
Mm-hmm.
I have a question.
Sure.
Yeah, I was wondering, I don't know if it's a dumb question or not, but how did you go about getting samples for your research? Like, did you go out and find samples, or did you have to go about it a different way?
So I was fortunate in that my advisor, Dr. Levitt, has a wealth of genomic data that he has gone out and collected over his career, just like thousands of lichens. And so he has a great library of these sequenced lichens. And so I was able to use the data that he had generated over the course of his career, which is I'm really grateful for. But most of them are from, and I forgot to mention this, but most of them are from the West. So Idaho, Wyoming, Utah, so in like this general area. But yeah, so I was able to use the data that he had generated. But that's a great question.
Hi, so I was just wondering, you talked in your presentation earlier about how in like intro bio classes, hybridization is taught as like a dead end. You like use the mule example and stuff. And from your research, it's, I don't know, you've shown that hybridization can play a huge evolutionary role. And I was just wondering why it's not taught in intro bio classes that hybridization can cause that effect. Is this like a new concept that just is not fully understood yet, and so that's why it's not taught, or is there a different reason?
That is a great question. I wonder that myself, as someone who has put this much time into researching this. I do wish that this is something that was taught like in introductory biology classes. I think a lot of it goes back to, so I've been reading this book by Dr. Michael Arnold about his research over the past several decades looking into hybridization, and he kind of you can kind of sense his frustration that it has taken so long for scientists to really start to accept that hybridization is something that is playing a role here. So I think in science, it takes a long time for us to, there's always this dominant thought, the dominant theory. And so I think it takes us a long time to accept these new ideas. And so I've found that there are a lot of scientists who really are still thinking that this is something that's not playing a role, but all the new research that's coming out is starting to show more and more that this is something that is important to look at. So I would love it if they started including that in textbooks, but I think it's going to be a while. I don't know. It takes a while for us to accept new ideas and really integrate them into our teaching. And so, yeah, I would love it if we could update textbooks.
I also had a question for you. Initially in your presentation, you mentioned that before we kind of understood what lichens were and how to classify them, there wasn't a huge economic purpose for them. But as we've come to understand more about them and how they behave, what would you say would be the biggest overall economic purpose that lichens are used for?
That's a great question. I think they still don't really have, there's no like industry that's working with lichens and working on, selling them, and they're not really integrated into, I would say, I don't know, they're not really integrated because we don't really know how to purposefully grow them, how to domesticate them and grow them. And so I'm not sure if they will, when or if they will become this like really big part of our economy. As I mentioned earlier, there's starting to be this trend where people are selling bags of lichens for like decor and stuff like that. And there are some cultures that use it for like they eat it, but I still don't think they are really, I don't know. I actually don't know if they're selling like bags of lichens that you can just cook up a soup or what, but I can't imagine if they will really have a huge economic purpose. And so, which is kind of nice because I think it can really do bad things to the population if it does kind of shift into like people wanting to go out and forage them and like destroying populations and stuff like that. So yeah, it's a great question.
So as you were analyzing your research and constructing the phylogenetic trees, were you generally surprised to see evidence of hybridization? Yeah,
that is a great question because when we went into this, we had no idea what we were going to see. Because when I started this, there weren't really any other studies where they had explicitly tested for hybridization among lichen-forming fungi. And so this was kind of just like a shot in the dark where we were like, well, we see this interesting pattern with the asexual vagrant lichens. So maybe we can just throw some of these hybridization tests at it and see if we can find evidence for this. And so it was really, really exciting, I guess, to see that there was evidence of hybridization. And I'm actually working on another, looking at hybridization among another group right now. And with this group, we thought, oh, wow. With the rock posies, we found really a lot of supporting evidence for hybridization. Let's apply this to another group. But in this other group, we're really, there's like a tiny amount of hybridization that we're detecting, but really a lot of it comes from how messy, maybe that's a conversation for another talk. But yeah, to answer your question, it was really cool to see evidence of this because it hadn't really been explicitly tested before.
I can go.
So earlier in the presentation, you talked about how there's not enough technology right now to further explore. I think it was maybe the evolution, certain aspects of the evolution of lichens. I can't remember what you were talking about, but like what kind of technology would be needed to further do that?
That's a great question. I think, so this, maybe I'm biased. I'm a little bit biased because I really truly think that the direction that we're going with our ability to sequence and analyze genomes is really, really revealing a lot to further our understanding of how evolution works and how that interacts with the genome. So from my biased perspective, I think that we're really in like just the beginning of understanding how that's impacting the genome and how to really analyze that. And so I really think I am really excited to see where the future takes us with looking into genomic data. I can't hear you, Anna. I think.
Can you hear me now?
Yes, I can hear you.
Oh, good. Yes. So thank you so much for this. Very interesting. Early on in your presentation, you talked about lichens being an affordable way to assess air quality. But they, of course, exist in some very polluted areas as well, such as on trees in city environments. So are there any specific lichens that are good indicators?
Yes, definitely. So it's definitely not like a generalized thing where you're just looking at all lichens. There are some lichens that are that we've found to be a lot more sensitive to pollution, and we've been able to understand how specific lichens respond to pollution. So in these biomonitoring studies, they often focus on specific species of lichen, looking at or like comparing ones that are super sensitive, and then we have the lichens that are not that sensitive at all. So yeah, that's a great question.
All right, thank you.
I got a quick question.
Sure.
So you said, and I don't really know how to phrase it, but you said that most lichens, I don't want to say belong to the group Ascomycota, but they're kind of derived from the group Ascomycota. Is there a reason for that you know of?
I don't actually know. Yeah, that's a really good question, but I'm sorry, I don't know the answer.
Oh, you're fine.
Thanks for the very interesting talk on lichens in general and then evolution in general as well. I just had a question about the terminology. So you talked about lichens as an ecosystem, and so that's species interacting in their physical environment. And I would think of a lichen as more like a community because it's an algal or a cyanobacterial component and a fungus. So tell me about that distinction, community versus ecosystem, when you're talking about lichens.
Yeah, that's a really great question and something that I think about a lot in my research. So there's, we're finding, and to shift again to like focusing on genomic sequencing, we're starting to develop something called metagenomics, which is you're able to capture the DNA that's there in the community that you're sequencing. So we do this a lot with like the human gut, looking at the microbiome in the human gut and what different bacteria are in there. And you can also apply this to lichens, looking at everything that is in there when you're sequencing it. And so we're finding that it's a really complex, so the symbiosis is generally between the fungus and the photobiont, but there can be multiple fungi growing in there. There can be multiple photobionts. And we're also finding that I'm sure a lot of people have heard about that recent study that came out looking at the yeasts and how that could be playing a role in the symbiosis. And we're also seeing that there's also a wealth of like bacteria that can be living among the lichen, but not necessarily contributing to the symbiosis itself. So it's a really interesting area of research looking at how this little, this little structure, this little symbiosis can be really hosting this like wealth of species.
Mm-hmm. Okay. Yeah, I'm just thinking if they're species though and they're living, I would refer to it as a community. But if you're taking into account the rock or the soil, then it becomes the ecosystem because it's species interacting in a physical environment. But if you're just talking about yeast and bacteria and algae and fungi, I would call that a community.
Okay. Well, I mean, I guess that you can make several arguments about it, but yeah, it's an important point to make.
Okay. Yeah, I just want to make sure I understood your point of view.
Mm-hmm.
Thanks.
Yeah.
Hi, my name's Joyce. I'm actually a current student at UW-Madison in Cons Bio, so that was really cool to hear. Hello. And I just took my introductory bio courses and did learn the whole, like, if they can't reproduce, then they're species and hybridization is bad. And so ever since you said, like, no, that's not super true, something's been very stuck in my head, which is how would you define a species then? Like, what delineates it?
That is such a great question, and that is an entire conversation, because that can be so. And I think you bringing that up is like really great because that's something that I like think about a lot, and it's something that a lot of scientists really debate about is like how can you really define a species. And so if you dig into it, you can find out that there's multiple definitions there's multiple species concepts, and that you're not really taught in school. And so I don't want to go into them all here because it's a long conversation, but there are multiple species concepts, and this is something that many scientists are debating, like how do we define a species? I personally have not decided on how you would accurately define a species, but I do think that the biological species concept is like far too reductive to really accurately define a species. So yeah, but that's a great question. And if you're interested in it, just Google different species concepts because it's really fascinating to see the conversation that surrounds that.
Awesome, thank you so much.
Yeah.
Hi, I had a question, I think, just about lichens in general, kind of going along with one of the previous questions. You said that there can be different photobionts or different types of bacteria or alga that are within the lichen. Are those interchangeable, or have they evolved with that mutualistic relationship to where they have to survive together?
That is such a good question. And unfortunately, I don't have an answer for you because that's something that we're still really actively researching, and we don't really know entirely. So sometimes, yeah, that's a really good question, and it's so complicated though that like I'm not sure I can answer it here because it's still something that we're really actively researching, and we don't entirely know how those different symbionts are really influencing how that lichen is growing. And there's been a lot of research looking at how the photobiont, so you'd have the same fungal symbiont, but if it switches the photobiont, it's able to grow in like a more extensive range. And so there's been a lot of research in that, but I think the research looking at everything contained within the lichen and how that influences its growth and everything is really something that we're really just starting to dip our toes into.
Awesome, thank you.
Mm-hmm.
Hi, my name's Aaron. I'm sorry, can you hear me? Sorry, the thing won't let me raise my hand for some reason, but basically, sorry, my question was just on just a piece of your research. You you mentioned you were studying vagrant lichens, which are primarily reproduce vegetatively, and you found like a ton of hybridization. How does a species that almost never reproduces sexually hybridize with another species?
That is a great question, and I'm really happy that you pointed that out because I'm sure other people had that same question. So the hybridization that we're looking at is further in the past. So it could have happened before they really evolved into like what they are today. But another thing that I would like to highlight is that sometimes with hybridization, something that you'll hear about is incompatibilities. So when it hybridizes, often the genome doesn't really fit, like the two genomes that are like combining don't really fit together as like neatly as you would expect. And so something that can often happen with those incompatibilities is a loss of sexuality. And so with our study, something that we kind of hypothesized after we looked at our data is that perhaps this asexuality developed as a result of this incompatibility, but that's something that we don't really know for sure. It's just a guess.
Thank you. That's really interesting.
Any other questions? All right. Well, thanks a lot for coming, and I'm really happy that so many people had so many great questions about this because, yeah, it's super fun to learn about, and I'm happy to teach you more about it.
Thank you a whole bunch. I really enjoyed your talk.
Oh, thank you.
Thank you.
Thank you, and happy Darwin Day.
Thank you.